what we have studied is that once the sample is collected, we do the cultivation, right? And cultivation can be done in a different way, having a requirement specific or the general in the anaerobic or the aerobic. Once you cultivate the organism, you have to use the polyphasic taxonomy for the microbial identification, and that polyphasic taxonomy can include your numerical analysis, your morphology, your physiology, right? Chemotaxonomy, typing and analytical method, and also genotyping based on the DNA, RNA, or the protein-based methods, right? Uh, so we paused for a moment to go inside the genotypic method, and then we explained the imaging techniques. So in imaging techniques, we have seen that it has started from a single cell, and then now it has reached up to a very certain, a very high quality imaging techniques using the electron microscopy and a scanning probe microscope. I have also explained you about the paper microscope, that is a foldscope, and you can have this demo in the field. I will bring my foldscope, and also showed you some of the videos where how the high resolution microscopes are used in the understanding the ecology of the marine microbes. Also, imaging techniques was further utilized by the epifluorescent microscope through the fish and cardfish, where you stain the molecular parts like DNA or RNA, right? And then you use the similar epifluorescence methods in the flow cytometry, and that helps in getting not only the enumerating the cells in the environment, but also helps you to get the single cells, right? That can be utilized again for single cell genomics. Now, coming to the molecular method, in the last 20-30 years, we have seen a considerable increase in the utilization of the molecular techniques to identify or to enumerate the microbial cells, and that includes PCR techniques, that is polymerase chain reaction, and first generation sequencing, that is applied biosystems or Frederick Sanger method, that automated DNA sequencing, and then developed to second and third generation, that is Illumina 454, and then to PacBio and Oxford Nanopore sequencing, right? In the molecular method, we have seen that the first step is to collect the sample, and then we do the DNA extraction. After the DNA extraction, we can directly go to the sequencing, that forms a part of the environmental metagenome. That shotgun metagenome sequencing. And if you are going for a specific reason, that can be called as a targeted metagenome. In case of bacterioplankton community analysis, we generally do with the 16S rRNA gene sequencing. If you want to go for a functional based metagenome, then you can choose the gene either for any antibiotics, for any enzymes, or for example, I give you for ammonia monooxygenase gene of RPA, right? So that is function based metagenome. Then we looked in detail that why we go for the PCR. So PCR is being done for the molecular markers. The reason which you sequence from the genome is called as amplicon, and that amplicon could be 16S rRNA gene in case of bacteria. We choose 16S rRNA gene because this 16S rRNA gene do have a number of variable regions and at the same time constant regions. So these constant regions are or also called as conserved regions, can be utilized to prepare the oligos or the primers, which helps to go and bind which we use in the PCR, which goes and bind to the conserved region and amplify the variable region. This variable region have the information about the species. It is unique for each individual or for each species, and that gives the understanding of the community, bacterioplanktonic community in the marine environment. One example I gave you from our study where we have cultivated the bacteria from the salt pans of Maracanum, and then we have obtained several different individuals of colonies. Upon sequencing and doing the bioinformatic analysis, we have found that there are three major groups. First is the Staphylococcus, Hello-Vibrio, and Chromohelobacter. And this phylogenetic tree can be seen in a very clear way that it distinguishes all three groups of bacteria with a very high statistical support. Okay. Then the second part was shotgun metagenome, where I said that once you get the DNA, you don't perform the PCR, you directly sequence it. It is a cumbersome process. The bioinformatics process is tedious. However, once you complete the bioinformatic process, you get a lot of genes that can tells you about the different microbial diversity. That also can tell you about the different functional or the pathways, genes involved in different functions or the different pathways. And this was first achieved in 2004 by a group of Craig Venter et al. Right, and this was a groundbreaking paper. As I said, that it has told that a number of unique microbial genomes present, different phylotypes present, and also after this expedition, we know that in Sargasso Sea we got the SAR-11, which is the most abundant bacterioplankton in the ocean, ubiquitously present across the world ocean, right? We also saw that there is advancement in the sequencing strategies, though I am not going in detail about how this sequencing works. What we need to understand that the first generation was a single sequencing. It is a high cost approach. I mean, it is costly approach. It is a more tedious approach in working in the lab, but it produces a very solid result, very good quality result. But if we want to do more and more number of genome, then we have to have a trade-off where we sequence small fragments of the genes and then combine through the cumbersome or tedious bioinformatic approach and then get the genome assembly, right? The third revolution happened, which is also called as third generation of sequencing, where you have the sequencers capable to make a bigger fragment, uh, and this particular Oxford nanopore is cheaper method than the Illumina or the PacBio, right? At present, most of the labs across the world has been using this Illumina, which is quickly moving toward third generation, because this Illumina is second generation, which is quickly moving to third generation. If the lab is very much well funded, then they are going for PacBio. Those who are doing more frequently, then they are moving toward Oxford nanopore sequencing, right? As a result, you can see that in last 25 to 30 years, from one genome in the prokaryote, we have more than 310,000 genome at present in the public database from the NCBI, which is being maintained in the by NIH in America. Okay, we have seen that after molecular methods, we have the inactive in situ activity methods also. One such method is the use of the microelectrodes and the biosensors. It's a very fine needle where all the reactions are happening at the tip. And this tip, once we are putting in the microbial mat, for example, or in the biofilms, or in the rocks, or any such certain in situ environment, tells you about the oxygen evolution or any other gases which is coming out from that particular location, which in turn tells you about the inactivity, I mean activity of the microbial community in the in situ conditions. Apart from that, we have the radioisotopes, which is now least used because it has been replaced by the stable isotopes. In the radioisotopes or stable isotopes, the primary idea is that if you label the carbon or nitrogen in the substrate, which is being utilized by the primary producers, thereby consumed by the primary consumer, tertiary consumer, and also by the if it is in between utilized by the microbes, that all you can analyze through the different autoradiography or the scintillation counters, right? Stable isotope is being developed, so several different probes have been developed, and at the same time, even the quantification has becoming more and more precise in terms of unique instruments in the mass spectrometry like nanosims, which has the capacity to find out the stable isotope within the cellular compartments, right? So not only the stable isotopes can tell you about the microbial functions and the food web studies, but also tells you about the cellular compartmentalization within the cell. Okay, this stable isotope has been also frequently used in order to understand the trophic interactions in the symbiotic associations in the marine environment. For example, between the chemosynthetic symbiosis, in the methanotropic symbiosis, in nitrogen fixing symbiosis, or the photosynthetic symbiosis. Last but not the least, the in situ activity has been studied using the mesocosm experiment, where the experiment has been done in situ in the marine environment, in a very big tanks like 1000 or 10,000 liters, and checking for their activity, whatever your study is. And then we have, of course, what we have seen, the utilization of the molecular approach in the form of microarray, metagenome, metatranscriptome, or metaproteome, in order to find out that what all functional genes or what all pathways are activated in that particular environment. So that tells you about the in situ activity as well. So this is what we have seen.